I'd like to welcome everybody to our MCHRI seminar series today on immune-epithelial interaction in pediatric IBD. Is type 2 good for you? I'm Elizabeth Bergener. I'll be moderating today, but I want to introduce our speakers. We have Dr. Michael Rosen, who's an endowed professor and director of the Stanford Center for Pediatric IBD and Celiac Disease. He's trained at Harvard, Boston Children's, and Vanderbilt. He joined us in the summer of 2021 after moving from Cincinnati Children's, where he was the director of their IBD center. He's a true translational researcher with experience leveraging samples and data from multi-center studies and heading a laboratory which studies mucosal immunology and epithelial cell biology, all of which is focused on improving the care of children with IBD. And then Dr. Babajide Ojo, who has a PhD in nutritional science from Oklahoma State University. He did a research fellowship at Cincinnati Children's under Dr. Rosen, and then also made the move to Stanford last summer, where he's now a postdoctoral scholar in Dr. Rosen's lab. And he has expertise on human colon organoids, which I think we'll be hearing about today. I'll turn it over to Dr. Rosen. If you guys have questions, you can put them in the Q&A, which I can help moderate at the end. So.
Thank you, Elizabeth. Let me take a moment to share my screen. All right. So before I get into the research presentation, I just wanted to talk a little bit about our Center for Pediatric IBD and Celiac Disease, which we're very excited about. Our mission is to integrate comprehensive clinical care with cross-cutting research to lift all children with IBD and celiac disease to achieve their potential. And this is a slide that I shared at Grand Rounds, but I wanted to share the high-level structure of the center and how it's structured. It's supported by, I should start by saying, it's supported by a really transformative gift, and that supports advancing our clinical care with faculty recruitments, allied health providers, and focus on training and patient education. But then, very relevant to MCHRI, a lot of support for research in the Stanford scientific community. So in addition to faculty recruitment, which is ongoing, we have both seed grants and research training awards that are administered through the MCHRI grants program, and refer you to the website. It's just Google MCHRI IBD celiac grants, that are generous grants, similar to other grants from MCHRI, but focus really narrowly on pediatric inflammatory bowel disease and celiac disease. And then we just have gotten provisional IRB approval for a biorepository, and that's really what we envision as linking the clinical care and research and building out a robust tissue and peripheral blood biorepository with associated relevant clinical metadata to support ongoing translational research and find new underlying disease mechanisms and eventually new treatments for these diseases. So it's a very brief sort of high level overview, but we're really excited about it and love to talk about collaborations. I'm going to talk today about research related mainly to ulcerative colitis, which is inflammatory bowel disease, or disorders of chronic inflammation of the intestine, whereas Crohn's disease is sort of the other half of IBD, can occur anywhere from the mouth to the anus. Ulcerative colitis generally occurs only in the large intestine to various extents from the rectum upward. You see on the left endoscopic image here a picture of a normal colon, and on the right a diseased ulcerative colitis colon lining or colon mucosa endoscopically. This is would likely be characterized as severe endoscopic appearance. You see complete loss of landmarks and vascular pattern in there, and spontaneous bleeding, and adequate degree of edema and erythema. And so, this is actually probably an underestimate, but from our best claims data, 15,000 children in the US have ulcerative colitis, and this is a rising incidence and prevalence. It's rising more so in children than adults. The initial treatment of ulcerative colitis is a combination of mesalamine, which is a 5-ASA anti-inflammatory drug, with or without corticosteroids, depending on clinical severity. And, but trials of suboptimal therapies, like when 5-ASA don't work or steroids don't work or patients become steroid dependent, leads to unfavorable drug side effects, particularly in the case of steroids, and delays remission of disease. So there's a need to individualize treatment approaches and predict response to therapy in pediatric UC. And there's still considerable morbidity. About 15% of children with UC will ultimately require a total colectomy. And although their surgical outcomes are good, this is still, I think most would agree, an undesirable outcome. And one big gap in our care is a lack of epithelial-directed therapies in ulcerative colitis. All of our therapies right now are really directed at the immune response. But mucosal healing, basically going from this diseased colon back to normal, which can be achieved in patients, albeit not as many as we'd like. Only about half of patients achieve mucosal healing. But that mucosal healing is the best predictor, not surprisingly, of long-term outcome. And of course, that requires restoration of epithelial homeostasis, as you would see in the image there. And so again, highlighting the need for therapies directed at restoring epithelial homeostasis. So I'm going to talk today about a more of a bedside to bench story where we begin talking about how we characterize the mucosal immune response and its relationship to outcomes in large cohorts of patients with children with ulcerative colitis, and how we used animal models to narrow down on the role of a specific cytokine related to the mucosal immune response observed in humans, and then move to even further reduction into enteroids, where we determine how immune cells interact with the intestinal epithelium to influence disease and homeostasis. And then Jide, a talented postdoc in my laboratory, is going to be following me and talk about our ongoing work with human collonoids, which I think is a work in progress, but we're equally excited about. So when I started in research, really, as part of my fellowship, there was increased recognition of this potential type 2 immune response in adult patients with sort of advanced ulcerative colitis requiring surgery. And some lines of investigation were implying it may be pathogenic. So a type 2 immune response is defined by the production of IL-4 and IL-13 by cytokines and innate lymphoid cells. By T cells and innate lymphoid cells. And as you see on the image on the left, the T cells, of course, express T cell receptors or antigen specific. ILCs, as many of you know, are part of the innate immune system, similar in their ability to secrete cytokines like T cells, but don't express the T cell receptor, so they're not antigen specific. And type 2 immune response is involved in the expulsion of helminths and the pathogenesis of allergic disease. And although there's been some implication of its involvement in adult UC pathogenesis, there's also been in recent years an emerging understanding of positive roles in tissue repair and epithelial differentiation. And so I determined that there were a few unknowns here. It was unknown whether a type 2 immune response occurred in pediatric patients with ulcerative colitis. It was also really unknown whether it existed in newly diagnosed patients prior to the onset, prior to therapies that, of course, modulate the immune response. So we had the opportunity to collaborate with the Risk Inception cohort sponsored by the Crohn's and Colitis Foundation. This was one of two landmark studies I'll discuss that was focused on Crohn's disease, but they enrolled 1,000 children newly diagnosed with IBD. And invariably, some of those also had ulcerative colitis, and they were followed to some degree. This is actually I think the only study that examined the ulcerative colitis patients in this cohort. And so we obtained rectal mucosal RNAs from a representative subset of patients in this cohort, nearly 200. Importantly, they were all treatment naive. Those with IBD had macroscopic rectal disease. And we used precise phenotypes specifically for Crohn's disease, where we divided the Crohn's patients with Crohn's colitis, only Crohn's. And these are difficult to distinguish from UC patients clinically sometimes, and Crohn's ileocolitis. And then we used a targeted approach, and we used this microfluidic real-time PCR array called a tilde array, and we could assess expression of 24 genes in duplicate associated with type 1, 2, and 17 inflammation. And so this is a subset of the targets that we looked at, and I'm going to focus in on a few here, but to orient you to the graph, red is the ulcerative colitis patients, blue is the Crohn's colitis patients, and light blue is the Crohn's ileocolitis patients. White is the non-IBD. And so if we zero in on some of the few cytokines of interest here, we found that there was indeed specifically increased expression of type 2 cytokines, namely IL-5 and IL-13, in UC really exclusively compared to even colon-only Crohn's disease. We also found there was increased expression of IL-13 receptor alpha-2. This is a decoy receptor for IL-13 that inhibits its bioactivity. Although this was in more of a stepwise progression, it was increased in ulcerative colitis more so than Crohn's disease, and specifically more so than colon-only Crohn's disease. And so we could actually, and in the interest of time, I'm not going to be sharing the data here, but we could develop a model with IL-5 and IL-17 actually that this could help distinguish ulcerative colitis from colon-only Crohn's disease in children. But we were very interested in a relationship with outcomes. And so this is a heat map of the gene expression in this subset of patients. And the UC patients are again in red, as noted across the top bar. And most of the UC patients, they actually clustered together the most, fell on the left side of this heat map in what we call clusters 1, 2, and 3. And that was really, those clusters were really defined by detectable expression of IL-13. And we found that UC patients specifically in that cluster had a much higher chance of 12-month clinical remission compared to those in clusters 4 and 5, suggesting that this type 2 immune response was perhaps associated with a superior response to treatment. And there are some limitations, though, I should note about this analysis, in that it's a relatively small number of UC patients, 40 to 50, and the treatment was heterogeneous. So although most were not treated with anti-TNFs, there was 5 ASAs, steroids, thiopurines in the mix there. So to overcome some of those limitations and to validate our findings, we had the advantage of moving to another landmark cohort to collaborate with called the Protect Cohort. This was a follow-on to the risk cohort. It enrolled 400 patients who newly diagnosed, this time specifically with ulcerative colitis. And as a sort of advance forward, these patients were treated with a standardized treatment approach, an algorithm of mesalamine with or without corticosteroids based on initial treatment severity. The very confusing graph on the right is the algorithm that I won't go into detail here, but just to show that there was a systematic treatment approach for these patients. And this study, a subset of patients, in this study, they did develop clinical predictive models for the primary outcome of corticosteroid-free remission without escalation beyond mesalamine. The model is noted on the right, but what I will highlight is that it does require knowledge of clinical status at week 4. So as the model as it stands, you would have to wait until 4 weeks of treatment to understand patients' likelihood of being in remission at week 52 on 5-ASA only. So due to the size of this cohort, we want to take advantage of that and look at some various responses to sort of specific treatment channels within the cohort. So we looked at 313 patients with available RNA within the cohort and looked at the primary outcome for everyone, which was corticosteroid-free remission on 5-ASA. But we also looked at those patients treated with 5-ASA alone and examined what we call corticosteroid-naive remission, meaning that they achieve remission on 5-ASA without being exposed to steroids. And then those subset of patients that had moderate to severe disease at diagnosis and looked at the likelihood of escalation to infliximab or a colectomy within 1 year. And so this slide gives an overall summary of our findings in the interest in time. I won't get into the details of all the models we developed. And so across the top are the outcomes of corticosteroid-free remission, corticosteroid-naive remission, escalation to infliximab, and colectomy. And then the first two here are sort of positive outcomes. And so the positive, what I have here is that the genes associated with the positive outcomes are on this top green row, and the genes associated with the negative outcomes are on this bottom red row. And the pattern we find, though, is that the type 2 effector cytokine IL-13 and instructive cytokine IL-33 were both associated with positive outcomes. IL-13 was actually, it seemed that was specific to the 5-ASA response and associated with corticosteroid-naive remission in those just treated with 5-ASA. And IL-33 was associated with lack of escalation to infliximab in those with moderate to severe disease at baseline. Whereas IL-13 receptor alpha-2, which, as I mentioned, depresses IL-13 bioactivity, was associated with negative outcomes for two outcomes, corticosteroid-free remission and escalation to infliximab, which is an anti-TNF medicine and a next line therapy. So type 2 cytokine activity here, again, appears overall to be associated with superior clinical outcomes. Another very interesting finding we had was that ROR-C, which is a canonical Th17 transcription factor, I didn't share with you the data, but ROR-C higher expression was actually seen in controls compared to ulcerative colitis patients. It was inversely associated with endoscopic severity. And here, higher expression was associated with protection from colectomy. And this is interesting because, the type 17 cytokines, IL-17, IL-23, are all certainly increased in ulcerative colitis. And we would expect potentially opposite effect. And we'll get into that in a few slides later. So this is just to come back to one model that shows you that if we did add IL-13 receptor alpha-2 to various clinically available data to predict week 52 clinical remission, we could come up with a model that was very similar in its accuracy to the Protect clinical model, but that didn't require that week 4 clinical information. So the addition of IL-13 receptor alpha-2 allows us to predict without waiting to week 4. A subset of the patients in the Protect cohort had available RNA sequencing data. And so as another approach, we performed whole genome co-expression network analysis, which identifies in an unbiased way co-expressed gene modules, and you can associate those with phenotypes or outcomes. And so one of the modules that pulled out, the black module, we found included IL-13 receptor alpha 2 as a hub gene. And so this gives us an idea of what biologic processes IL-13 receptor alpha 2 may be related to in this disease. And we see inflammatory response, cytokine activity, but also extracellular matrix, collagen binding, and wound healing. And another module that we call the brown module included ROR-C. And we see that this module has, within its associated processes, included lipid metabolic processes and cell structural processes like regulation of cell projection, regulation of microvillus assembly. And there are some genes here that are often associated with the epithelium, KLF5, CFTR, HNF4A, and others, potentially identifying an epithelial signal here. And so to understand what, where even better, where the ROR-C and IL-13 receptor alpha 2 and their co-expressed genes, what cell types are driving these signals, we turn to an established single cell data set in UC patients, and this is adult patients from a paper published by Smiley and colleagues in Cell. And so this diagram shows expression of on the top IL-13 receptor alpha 2 and its 20 most correlated genes in our data set, and ROR-C and its 20 most correlated genes within its co-expression module. And then we have various stromal cells, epithelial cells, and immune cells across the top. Now, this paper did identify IL-13 receptor alpha 2 inflammatory fibroblasts as a potential disease-involved cell type in ulcerative colitis. So not surprisingly, we found that from the data from our cohort, IL-13 receptor alpha 2 and its co-expressed genes are predominantly expressed in inflammatory fibroblasts within the stromal cell compartment. But perhaps even more interesting is what we found with ROR-C. And we found that ROR-C and its co-expressed genes were actually not predominantly found in the immune cell compartment, but rather in the epithelial compartment, and in particular, the less differentiated cell types of stem cells and various transit amplifying cells. So takeaways from these patient studies is that in a large treatment-naive pediatric UC inception cohort, targeted assessment of mucosal immune gene expression predicted clinical outcomes, and that a model including IL-13 receptor alpha 2 expression and only baseline clinical factors was accurate for predicting corticosteroid-free remission as the original model that included week 4 remission data. The association of type 2 immune genes with improved outcomes and the type 2 counteracting gene IL-13 receptor alpha 2 with poor outcomes implies a potential beneficial role of type 2 cytokines in UC. And so we feel these findings support further explanation of IL-13 receptor alpha 2 as a potential therapeutic target in UC, and certainly future studies of the epithelial specific role of ROR-C in colon mucosal homeostasis and UC pathogenesis. So I mentioned that interleukin 33 is a type 2 instructive cytokine, and it's an IL-1-like cytokine. It signals through its receptor ST2. And IL-33 is well known to induce type 2 cytokines from T cells and innate lymphoid cells. And it's been established that it's increased in the colon mucosa of adult patients, and we've shown it's increased in pediatric UC as well. So we were interested in understanding the role of IL-33 and IL-33 signaling in ulcerative colitis, and to just with ixazolone in both IL-33 deficient mice and mice deficient for the receptor ST2. And what we found was kind of contrary to our initial hypothesis that IL-33 may be driving disease. We found that mice deficient for IL-33 or its receptor had increased histopathologic severity, as shown on the left. And this was associated with really a profound depletion in goblet cells, as shown in the PAS staining on the right. Now, intestinal mucus helps maintain barrier function. The inner mucus layer is largely impervious to bacterial penetration. And mice lacking MUC2, the principal intestinal mucin, have increased bacterial translocation, and they develop spontaneous colitis. And so to further dig down into how IL-33 may be regulating intestinal goblet cells, we moved into a primary enteroid culture system. So one can make enteroids or collonoids from tissue stem cells from mice or humans when they're cultured in the appropriate media with the appropriate growth factors. And we can change these growth factors to reduce the Wnt in these in the system to drive differentiation, where you get these three-dimensional structures that have all the differentiated cell types of the epithelium and have structural domains similar to the epithelium, such as crypt domains and villus or surface epithelial domains. And so we exposed murine enteroids to IL-33 directly. And we actually found, this is UEA1 staining, we didn't see any increase in goblet cell numbers by exposure to IL-33 directly. We did, not surprisingly, see a large increase in the number of goblet cells with exposure to IL-13, and that's been demonstrated by others in other systems that IL-13 induces goblet cells. So it doesn't seem like IL-33 could regulate goblet cells directly in the epithelium, and probably needed some intermediary cell type. And to summarize a number of experiments here, we created this co-culture system where we could co-culture enteroids and a transwell over various cell types. We tested immune cells and fibroblast cells. But in the end, we found that when we generated CD90 positive mesenteric lymphoid cells that were enriched for predominantly ILC2s by injecting mice with IL-33, and we exposed this system to IL-33, that's when we were able to induce goblet cells in the epithelial enteroids. And so in other work, we've shown that, at least in mice, IL-33 during colitis seems to be coming predominantly from actually fibroblasts, but it acts on ILC2s and potentially Th2 cells to induce the production of IL-13 that acts, and we demonstrated through transgenic enteroids, acts on IL-13 receptor. We demonstrated the requirement for that to induce goblet cell differentiation in the intestinal epithelium. And now we're excited to be advancing this to human systems, and we've developed methods for enriching ILC2s from human blood and expanding them ex vivo, and growing collonoids from our patients, and can use the same type of system to co-culture them together. And this is preliminary unpublished data, but we do see that using when we co-culture the collonoids with the ILC2s, we see increased budding, which is a measure of differentiation, increased expression of MUC2 associated with goblet cells, and sort of decreased expression of LGR5, the major stem cell marker for intestinal cells, epithelial cells. So what I've shared today is that in patients, we've shown that molecular tissue gene expression data can predict outcomes and identify new targets for research. In animal models, we've shown that the cytokine, the type 2 instructive cytokine IL-33 is protective in acute murine models of colitis and preserves goblet cells. And that in intestinal aneroids, we've shown that IL-33 induces goblet cells via IL-13 producing ILC2s. And so I'd like to acknowledge the current growing laboratory at Stanford, and excited for get G-Day to share with you ongoing work with human enteroids. My recent laboratory members at Cincinnati, Amanda Woodell and Cedral Fox, work together on the co-culture methods. Amanda has led our murine work and immunology work. The risk and protect investigators, there's really 30 of them at least, and their PI leadership, and then collaborators at Cincinnati and others, and particularly Rebecca Carnes, who's done a lot of the statistical and bioinformatics work with the cohorts, and Anil Jaga and Mahika Sharma, who collaborated for the WGCNA analysis and the single cell analysis. And of course, our funding from NIH and Crohn's and Colitis Foundation. And so with that, I think I'm thrilled to pass the presentation on to G-Day. I will stop sharing.
All right, let me share my screen. All right, so I'm excited to talk to you today about what I've been doing for the past 1.5 years in Dr. Rosen's lab. And I appreciate the opportunity to really share this with you. And I'll be talking about, patient-derived colon organoids as a preclinical model of pediatric ulcerative colitis. Now, today, I'm just going to share ongoing work, really. And I must say right off the bat that most of the things I'm going to share today are things we are still working on, but we are just going to share what we have and what we are thinking in terms of the broader potential of collonoids, as I will call them, for the rest of this presentation. Now, as Michael said earlier, there is really a lack of human colon epithelial models for preclinical studies, and most of the models that we have right now are mostly focused on suppressing immune cells in the lamina propria, and there are a few ones, there are really none that are focused on healing the epithelial cells. Of course, we have immortalized cell lines, but they do not fully represent, the human colon epithelium, and mostly the genomic manipulations they go through make them less susceptible to certain environmental stimuli, which we know are important in the pathogenesis of ulcerative colitis. Now, of course, collonoids, organoids generally were reported as a potential model for many diseases, and I think that was in 2009 by the Hans Cleaver's lab. And really, you can, it means we can take tissues from animals and we can culture either the crypt, or we can dissociate them into single cells, and we can culture these in certain extracellular matrix. And the common one is Matrigel, and we have BME2 right now. And in that medium, with several growth factors, we can have self-renewing organoids that recapitulate a lot of the structural features of the original tissue. Of course, not just structural features, organoids have several advantages. Of course, they are primary cells. They can be primary cells because they can be derived directly from patients, which in theory, they should be able to remodel or recapitulate several distinct features of disease. And of course, you can cryopreserve them and use them as a long-term model of preclinical model of disease. And as we demonstrate in this figure, we can see MCHRI, which is a marker of goblet cells. We can see that these collonoids can recapitulate even how goblet cells are kind of squished between epithelial cells, as we often see in epithelial cartoons. But really, there is still a prevailing gap when we talk specifically about UC, especially in children. Do these patient-derived colon organoids carry forth all the associated molecular and functional features of the original tissue, just apart from structural features? And when we talk about molecular features, some of the things we often think about is, all the epigenetic signals or, transcriptomic signatures. But what we can also do is to look at what we call gene regulatory networks, which really offers higher magnitude inference of chromatin regulation. And by that, you can combine, like chromatin regulatory data like ATAC-seq or ChIP-seq with gene expression data, and you can use that to decipher how transcription factors regulate gene expression on the chromatin. So you can really look at associated transcription factors and how they drive a particular disease. So one question that is still lingering is, are these carried over in UC collonoids? Are there shared and unique, interactions among these GRNs, as I will call it in this presentation? Of course, we have the ability to, if these are going to be a good model, if collonoids would be a good model, then we should have unique features when it comes to GRNs that we would see in UC versus non-IBD. And we would expect that the tissue and collonoid GRNs will be similar. And when we talk about functional features, we are really thinking about, the metabolic function of these collonoids. Do they reflect like the original tissue? And when we think about this, we look at a recent report by Michael and his collaborators published this in 2019. And when they looked at the transcriptomic signature of the pediatric UC epithelium, they saw that a lot of genes involved in mitochondrial function of these epithelial cells are significantly reduced. And we know that the function of the mitochondria is vital for differentiation of the epithelium and replenishing the epithelial barrier. And you can see here that certain important proteins in complex 4 are significantly reduced in active UC. So a question we can ask is, are these collonoids really reflective of the original tissue when we look at the metabolic landscape of these collonoids? And what environmental stimuli can induce or reinduce these phenotypes? So part of what I'm doing kind of looks at two broad aims. First, we would like to define disease-associated gene regulatory networks in pediatric UC, and really to see if these patient-derived colonoids recapitulates these GRNs, and also look at if these colonoids reflect the mitochondrial impairments that have been reported in UC, and if there are specific environmental stimuli that impairs or helps reduce these phenotypes in colonoids. So a study design, just a summary of our design here. We have been recruiting non-IBD and UC volunteers for the past, since we started at Cincinnati. And we take the biopsy and we kind of split that into two. So we take some of them and we extract colon epithelial cells, which we deplete CD45 from this. And from these primary CECs, we take a portion for RNA extraction, and also a portion for epigenetic analysis that I will talk about soon. And also the second portion of the biopsy, we convert that into colonoids that we have been freezing down in biorepository. And also we can take these colonoids as well to assess mitochondrial function, and also take a portion of this for RNA extraction and post-translational modification analysis here. So basically, we can combine the RNA-seq and epigenetic data to define differential GRNs either in UC versus non-IBD, and also in a primary colonoids and in primary CECs rather, and colonoids, to see if these colonoids actually reflect the molecular landscape of the original tissue. So of course, like I said earlier, we expect that theoretically we would expect that the GRNs that are specific for pediatric UC in the original tissue would remain persistent in the colonoids that we grow from this tissue. And one of the first things why we are thinking about this is one of the first things we have done is to just when we just took some of these colonoids and we stimulated them with flagellin, which is a pathogen associated molecular pattern of bacteria in the gut. So usually our epithelium is exposed to this, and at lower concentrations, you are not supposed to have detrimental response to this. But when we look at UC colonoids, we can see that the IL-8 gene, which codes for IL-8, that is a strong chemotactic factor for neutrophils and T cells in the gut, is heightened. The expression is like at least over fourfold increase over other groups. So which shows that there are certain molecular signals that makes UC colonoids more susceptible, or UC epithelium more susceptible to environmental stimuli. And we believe with this analysis we might be able to uncover those specific control points. So one of the methods we've been optimizing in the lab is cleavage under targets and fragmentation, which was pioneered by the Ennicoff's lab in 2019. And really it's similar to ChIP-seq, but it's similar to ChIP-seq in the sense that it's an antibody-guided assay, but it's superior because it requires less samples and gives less noise. But basically, when we isolate the nuclei from these epithelial cells, we add a primary antibody to a specific target, and we add the transposase enzyme, which is tagged with protein A and G and protein G, which helps to guide this transposase enzyme towards the Fc receptor of the antibody that we are targeting. The cool thing about this is the TN5 enzyme already has Illumina sequence adapters So as soon as you activate the enzyme, it makes specific cuts at that region of interest and ligates the sequence adapters to that sequence, and you can just take that and amplify and sequence without doing that. You omit the library preparation step, as you would often do with ChIP-seq. And for us, we are more interested in the histone H3 of the chromatin by looking at monomethyl at lysine 4, monomethylation at lysine 4, which is a marker of poised enhancers, and also trimethylation at lysine 4, this H3 histone, which is a marker of an active promoter, and also acetylation at lysine 27, which is a marker of active enhancer. Now, what we have found in preliminary studies is that when we take one patient sample and we looked at both the primary CECs and the collonoids, we see that these histone marks are really carried over into the collonoids, and we are looking at the FOXA2 locus here, which regulates goblet cell differentiation. And I just highlighted ME3 here, like I said, the marker of active promoters, right? So this gives us encouraging signs that we will be able to annotate, differential regulatory sites on the chromatin when we compare UC and non-IBD collonoids, and also primary CECs and colon organoids. However, one of the challenges we have run into is for downstream analysis of these GRNs, we would need a really high number of unique fragments or unique reads. And we, at the moment, with our cut and tag analysis, we are seeing a high redundancy of fragments. Now, assuming we just want to look at the big read files and look at peak, the peaks as I showed in the previous slide, we will be fine. But because we have to do the GRN inference, we would need a robust sequence, unique sequences for that. So, and we think it's because we have really insufficient samples, especially in our primary CECs. And what we are optimizing right now, working on right now, is using ATAC-seq. Now, ATAC-seq is not an antibody-guided assay, but it still uses the transposase enzyme. And the enzyme specifically recognizes open chromatin regions on the chromatin, which in theory we would get peaks that correspond to transcriptionally active regions of the chromatin. Now, we didn't consider this at the start because of the MCHRI that we use to grow these collonoids. There are reports that MCHRI contain cell-free DNA, which if we use ATAC-seq, we might get some spurious DNA sequences in there. But talking to our collaborators and the pioneers of ATAC-seq, they have recommended an improved protocol that we can hopefully use to reduce the cell-free DNA in the MCHRI. So that's what we are working on right now in the lab to see if we can proceed with this method. Now, we are hoping we will be able to get sequencing data from ATAC-seq and RNA-seq data to use combine these two by the help of our collaborators for GRN inference in these samples. Now, the next thing we are trying to do is to see if we can decipher differences in mitochondrial function in UC and non-IBD collonoids. And for that, we are taking advantage of the CEOS extracellular flux analyzer. And basically what this particular instrument could, when we played ourselves into these 96 well plates, and we challenged them with specific inhibitors of mitochondrial respiration, oligomycin, FCCP, and rotenone, antimycin A, the equipment can measure oxygen consumption rates in the media of these cells. So how these cells are responding to these specific inhibitors and are making use of oxygen. And also, it can also measure the acidification of the medium, which will correspond to glycolysis rate. But really, what we have done is to, optimize this procedure using, different methods, and we've been successful at doing this, considering that we have to play with MCHRI gel and all that. But we've been successful at doing this, and you can see here that when we stimulated this collonoid with different concentrations of LPS, which is another bacteria molecular pattern in the guts, and TNF alpha, we can see distinct reactions when we look at basal respiration here, and also maximal respiration. So at even the smallest concentrations. So this is something we are still gathering data on, but we are really excited to move that forward. Another way we are trying to assess mitochondrial function in these collonoids is looking at mitochondrial membrane potential. And we know that this is important for driving ATP synthesis in the mitochondria. And we're using the JC1 dye, which is a really cool dye when you look at how it functions. This dye usually emits green fluorescence, but when it enters into the mitochondria, it forms aggregates that emits red fluorescence. So we usually look at the red to green ratio, or fluorescence intensity to, determine which mitochondria might be healthy and which might be unhealthy. So one of the things we have done is to, take some of these collonoids and just stimulated them with flagellin, like I said earlier, which is a bacterial mouth molecular pattern. And we saw a difference when we looked at the red to green ratio. A higher ratio would signify a more healthy mitochondria, and we saw a differential response in them. You can see that you see it's kind of reduced, and when we looked at percentage change from untreated collonoids. So which shows that metabolic fitness of the UC is lower than collonoids. So we are really excited to move this forward by testing more samples, and we are very excited to see what this would result into. So to sort of conclude, we believe, studying these patient-derived organoids and establishing control points that we can see in collonoids will help us push forward potential interventions that can be directed towards the epithelium to improve mucosal healing. And this could be important for, high throughput drug screening and personalized medicine in pediatric UC patients in future. And I would like to thank a lot of people that have been pushing this project, and I think we'll be ready to take questions now.
Great, thank you for a wonderful talk. It was very, very interesting, and you have some good questions coming in. So the first couple questions are for Dr. Rosen specifically, and Alexa Weingarten is asking, IL-33 and ST2 are also involved in Treg function. Is it possible that the increased inflammation is related to altered Treg function in your knockout mouse model? Oh, you're muted, Dr. Rosen.
Yeah, it's an excellent point. It is possible, and we we did, I'm trying to remember if we have to look back to see whether we studied, I don't remember there being major differences in IL-10 production, although of course it may be, like for instance, IL-33, as I'm sure you know, Alex, has been implicated in Treg production of amphiregulin, which could also have effects on the epithelium. So we weren't able to answer that question, but I think it is potentially a possibility.
Great. And our next one we have from Dr. Melissa Mavers. It's a little bit longer. Thank you so much. This is exciting work. I focus on invariant natural killer T cells in graft versus host disease. And we recently published a paper showing that the adoptive transfer of Th2-like and similarly surprising Th17-like invariant natural killer T cells were protective in GVHD. Interestingly, we found that via ATAC-seq that INKT2, but also INKT17s have increased chromatin accessibility at the IL-4 promoter and induced changes in conventional T cell transcriptomes, including downregulating of IL-18 pathway and tissue homing integrins. I was wondering if you've looked at IKT cells in your mouse model or in your models, and also whether you've looked at similar impacts on conventional T cells.
No, thank you. That's a lot to unpack, and I'd love to talk more. Originally, you're probably aware that the IL-13 producing cell in mice with oxazolone colitis was identified as an invariant natural killer T cell, and then in humans it was identified as an atypical NKT that didn't have the invariant receptor. We haven't been able to sort of reproduce those findings, and I think it's probably a broader cell type that's producing it. And so we had begun to look in the mouse. We haven't in the humans just because the original work sort of already determined that it was an invariant NKTs producing the IL-13. But I'm sure the field on invariant NKTs has progressed tremendously since I last dug into it on that, and I'd be happy to talk more, definitely.
Great. And then we have one from Dr. Oro as well. In patients with skin psoriasis, IL-17A/23 feed forward drives inflammation and barrier breakdown, not unlike IBD, but they appear to worsen in IBD in trials. Do they have a negative effect on IL-33 and IL-13?
Should have a negative effect on IL-13. My guess is it's probably the IL-17, in IBD, anti-IL-17 didn't work and potentially made it worse. And then obviously now we're seeing patients treated with anti-IL-17 for rheumatologic and skin conditions, and they develop IBD. So there is this problem with IBD, with so many of the things that are driving inflammation also have an effect, I think, on a positive effect on epithelial barrier function and proliferation. And so what may look like it's good at addressing in mice, the balance may be differentially affected in humans. So I think it's probably due more to epithelial effects, but I think that's another potential alternative hypothesis. Of course, blocking IL-23 in IBD seems to be very effective. And that's a whole nother very interesting question. Why is blocking IL-23 effective and blocking IL-17 not? What are the IL-17 independent mechanisms by which IL-23 is acting? And that's an area that people are interested in exploring.
Great, thank you. A complicated system and model, but it sounds like you guys are kind of drilling down on it. I think the organoid stuff is really interesting, and I'm a peds pulmonologist. We work with both cell culture and organoids and CF, and really use them for kind of serotyping or personalized medicine. It sounds like you guys ideally would like to get to that at some point in IBD. Is there, at this point, what would you predict would be a good readout in a collonoid for a patient? Like what, I mean, I guess that's part of what you're probably trying to figure out here, but we're lucky in CF, we know exactly what molecules affected and we can look at the chloride conductance. But is there anything like that in IBD that you think could be that readout?
Jide, you want to take a stab at this?
Yes. Of course, it's, like you said, it's part of what we are still working on to identify those control points. But if I would think about it right now, I would say an ideal readout would be, factors that increase things like goblet cell or MCHRI regeneration or proliferation in our organoids. So that's, I think, would be something that might be, feasible to try to be translatable in humans. So that's one of the things we can think of right now. But I'm sure we would identify some other control points when we are done with the study.
Great. That sounds wonderful. I think that's all the questions that have come through. Thank you to everybody that attended today. And thank you so much for these wonderful talks and sharing your research.
Thank you. I encourage anyone to please reach out directly with ideas or collaborations, or if they have questions about the center, grants, etc. So thanks so much for everyone's attention.
Thank you for having us.